Hello everyone and welcome to my channel AVpedia. In today's lecture, we are going to study about phylogenetic classification and how to construct a cladogram. Now, phylogenetic classification is the classification which is based on evolution. This type of classification was given by Carl. On the basis of phylogenetic classification, the classification of the organisms are divided into 6 kingdoms. And this classification was given by Carl Voss in 1991. According to Carl Voss, the organisms are divided into 3 domains, and these three domains are bacteria, archaea and eukarya. A domain is basically the largest taxon of classification, which is further then divided into kingdoms. Bacteria, according to the 5 kingdom classification, there are 5 kingdoms of classification, in which one of the kingdom is Monera. In the 6th kingdom classification, the Monera has been divided into 2, that is bacteria and archaea. Archaebacteria includes those bacteria which are found in very harsh conditions, whereas bacteria includes all the normal bacteria which are found in normal conditions. And eukaryotes include all the eukaryotic organisms. Now, when this type of classification is done on the basis of evolution, then this type of classification is also known as a 3-dimensional classification, and it is a vertical classification. When we go for the phylogenetic system of classifications, then it results in the construction of phylogenetic trees. So in order to construct a phylogenetic tree or a cladogram, first of all a Venn diagram is created by which we group the organisms on the basis of the similarities and dissimilarities in their characteristic features. So for example, now here there is a common area where all the 3 domains are overlapping. So this common area, this is known as LUCA, that is lowest universal common ancestor. Now this LUCA is having those characteristic features which are common in all the 3 domains. Now these characteristic features can be, for example, all these organisms, they are made up of cells. They have got a cell membrane. They have got DNA, RNA, ribosomes, cytoplasm. Now these characteristic features are common in all the 3 organisms, and that is why viruses have been excluded from this, because viruses are acellular structures. So they are not included in this group. Now when we come to the differences between bacteria, archaea and eukarya, then first of all the bacteria are having prokaryotic nucleus, and archaea are also prokaryotic, but eukarya are eukaryotic in nature. Now we come to the cell membrane. The cell membrane is made up of phospholipids. Phospholipid is a structure which has got a head and a tail. Now in bacteria and in eukarya, this phospholipid layer is a bilayer. That means 2 layers of phospholipid molecules are present, and the bonds which are present here, they are ester bonds. But in archaea, there is a slight change in this phospholipid layer. The phospholipid layer which is present in the archaebacteria, they are branched. And because of this branching, the two phospholipid layers, they are continuous with each other, so that there is a monolayer of the phospholipids. Another thing, that they have got ether bonds instead of having ester bonds. So on the basis of this, the prokaryotic organisms have been separated from here. Now, another thing is the cell wall. The cell wall which is present in bacteria, it is made up of peptidoglycan, whereas in archaea, in eukaryotes, the cell wall is made up of cellulose, and in case of archaea, it is made up of murine structures. So, though the archaebacteria have got these modifications because they have to survive in very harsh conditions. Now, on the basis of these characteristic features, a cladogram, a graphical representation, can be constructed. So, this graphical representation is consisting of a cladogram. Now, here the common ancestor here, this is the common ancestor, which is LUCA, lowest universal common ancestor, and this portion of this cladogram is known as a root. Now, these cladograms, they give you the evolutionary relationship between these organisms, and those organisms which are not very related with each other, they are regarded as an outgroup. The common point is known as the root, whereas the next branches, they are known as the nodes. And the line on which every organism lies is known as a clade. And they are built on the basis of the keys which are obtained by classifying these organisms. Now, this cladogram can be constructed by the help of observing the structures of the organisms, or it can be done by genome sequencing, by DNA, RNA, homology, etc. Now, we take the example of a hypothetical group of organisms which we have found. So, on the basis of this, we try to construct a cladogram. Now, in this cladogram, in this group of organisms, first of all, we see which one is the most odd man out. So, this is a square shaped structure F. So, first of all, it comes here, F. After this is a circle which is H. So, this comes over here, H. After this, there appears an I over here. So, this becomes D. After this, there is a mouth. So, it comes here, E. Then comes A, in which there is a tongue-like structure also appearing over here. So, this is A. And after this, there are certain antigens or protection or covering of the cell also coming here. So, this is B. And next is C, in which the mouth-like structure has disappeared from here. So, this is C. Now, these organisms, they are arranged on the basis of their evolution, but still this is just a hypothetical and a probable series of arrangement. It may be true, it may not be true also. Now, they are related like this. And this line, it represents time. So these organisms are the oldest one and these organisms are the newest one. Now we have to determine whether B is more related to A or B is more related to C. So it is quite possible that in B a retrogression has taken place, and due to some adaptive features the mouth has disappeared. So that is why we have kept C over here. Now in order to find out whether A is more related to B or C is more related to B, we have to further take into account some other factors also. So a cladogram is just a hypothetical arrangement of the species, and it is done in a very planned and a systematic manner. These cladograms, they can be like this or they can be like this. Now we take one more example, and that is of a few animals which we have found, like sharks, frogs, monkeys, kangaroo, parrot. So we observe certain structures in these organisms. So vertebral column, vertebra are found. We look for vertebra, so it is found in all these organisms. Then we see limbs. So limbs are found in all these organisms. Feathers, they are found only in parrot. Placenta is found only in monkey and kangaroo, and hairs are found only on the monkey. So accordingly we prepare a Venn diagram, and according to this Venn diagram we prepare this cladogram. So on this cladogram, the first structure which is going to come is the shark. Then the next structure which is going to come is the frog. Then parrot. Kangaroo. And monkey. Now here we will write those characteristics on the basis of which this classification is done. So here vertebra. So this the character which comes first, it is present in all these organisms. Now in frog, 4 limbs. So these 4 limbs are present in all these organisms but not in the shark. Then feathers. Then placenta. Then hairs. So this node here, it represents a common ancestor over here. Now these cladograms, they, as I told you earlier, that they are just hypothetical structures, but in order to construct, there are certain a phylogenetic tree is constructed which is based on actual age of the fossils, and it is more branched. And all these branches, they depict the relationship between the organisms. So farther the branch, more less related they are. Smaller the branch, more related they are. So in this way, a phylogenetic tree of life is also constructed. So this was all about the lecture. Hope you liked it. If you like my lecture, please subscribe to my channel and also download my app Teach Biology. Thank you.